Standard 12 Chemistry, Unit 14, Biomolecules, Part 9. Today we are going to learn about nucleic acids. The particle in the nucleus of cell, particles in the nucleus of cell, which is responsible for heredity, responsible for heredity. This particle is known as chromosome. The chromosome is responsible for heredity. Here, the chromosomes is made up of proteins and nucleic acid. Nucleic acid is long chain polymer of nucleotide. And the nucleic acid is also known as polynucleotide. Mainly nucleic acid two type, RNA and DNA. DNA deoxyribonucleic acid, RNA ribonucleic acid. Now the chemical composition of nucleic acid. On hydrolysis of DNA, RNA, we get pentose sugar. Number 2 is phosphoric acid, and number 3 is nitrogen containing heterocyclic compound. Nitrogen containing heterocyclic compound, they are called base. Mainly here we discuss 5 types of base. Number 1, adenine, guanine, capital G. Adenine and guanine is purine base, while cytosine, thymine and uracil, these 3 bases are pyrimidine base. Also important for MCQ also. Mainly 5 nitrogen containing heterocyclic compound, and they are called base. Adenine, guanine, they are purine base. Cytosine, thymine and the uracil are pyrimidine base. DNA and RNA that contains mainly the DNA contains AGCT, T for thymine, capital T, and RNA that contains AGCU, U for uracil, capital U. That's the main difference. Second thing is most important. In DNA, sugar moiety or sugar molecule is beta D 2 deoxy ribose, while in RNA is in DNA, while in RNA the sugar moiety is beta D ribose. They are pentose sugar beta D ribose. Here the 5 carbon chain 1 2 3 4 and 5. First carbon and fourth carbon attach with oxygen atom. And this is the structure of beta D ribose, and it is Fischer projection. Now the Haworth projection for beta D ribose. Here the 5 carbon, first and fourth carbon is bonded with oxygen atom. The first, second, third, fourth and fifth carbon. On fifth carbon CH2H and CH2OH. Now the position of the other group and atom, it will be on upper side and on lower side hydrogen, and here the on lower side OH, on third carbon OH group, and here the hydrogen and hydrogen on fourth carbon, here the hydrogen. Okay, this is the structure of beta D ribose. And it is Haworth projection. Now the structure of beta 2 deoxy ribose is also contain same structure, but the only difference here 1 2 3 4 and fifth carbon, these all are remains as it is. Here the OH and the hydrogen. Here only the difference, the oxygen atom is removed from the beta D ribose, and here the only hydrogen atom is present. This is the basic difference between two structure. Is beta D 2 deoxy ribose. It is Haworth projection. Now the next topic, that is structure of purine base. Mainly purine bases are adenine, the capital A, and guanine. Here the structure of adenine and guanine is similar to the purine. Here the structure of adenine and guanine, and here the structure of purine, that contains 1, 2, 3 and 4 nitrogen atom. 4 nitrogen atom. The numbering is 1, 2, 3, 4, 5, 6, 7, 8 and 9. Second thing is C5, H4 and 4. Its chemical formula is a structural formula. Now adenine, its IUPAC name is 6-amino purine. Here on 6th position NH2 group is attached, and the rest of the structure remains as it is. Okay. Then guanine, its IUPAC name is 2-amino 6-oxy 2-amino 6-oxy purine. So numbering start from here. The only difference, the H is first position, is second, third, fourth, fifth and sixth. On sixth position the oxygen group, carbonyl group, it is 6-oxy, and rest of the structure is remains. The guanine is 2-amino 6-oxy purine. This structure and this structure is nearly about same. The difference is here the hydrogen atom and the bond. Now cytosine, thymine and uracil, they are pyrimidine base. They are pyrimidine base, and here the purine base. Here the structure of pyrimidine is also nitrogen containing heterocyclic compound. Is first to second position, third position, fifth, fourth, fifth and sixth position. For cytosine, its IUPAC name is 2-oxy 4-amino pyrimidine. So on second position is carbonyl group, on second position is carbonyl group, and on fourth position is NH2 group. Then thymine, 2,4-dioxy 5-methyl pyrimidine. So on on is first, second, third, fourth, fifth, and sixth position. So on second and fourth, sixth position, a carbonyl group CO, and on fifth position, methyl group is attached. So this structure and this structure is only the difference. Second thing is bonding here. Then uracil, its IUPAC name is 2,4-dioxy 4-amino. So only the difference is here 4-amino. Sorry, here only a 2,4-dioxy pyrimidine also is here on first, second, third, fourth, fifth, and sixth position. On second and fourth position, a dioxy group, and rest of the structures remains as it is, 2,4-dioxy pyrimidine. Okay friends, this is the concept about the DNA contains 4 bases: adenine, guanine, cytosine, and thymine, and RNA contains 4 bases. The 3 bases are same, but the fourth one is uracil. Next time we will discuss the structure of nucleic acid. Thank you very much.